Hi everyone. In today's session, we're going to explore the Structure Activity Landscape Index, a powerful method that helps us understand how small structural changes in molecules can lead to big differences in biological activity. We'll start with a brief background on what Structure Activity Landscape Index is and why it's important for hit optimization in drug discovery. Then, we'll move to a hands-on demonstration using DataWarrior, where we'll analyze a dataset of thrombin inhibitors, the same dataset we designed in our previous session. Along the way, we'll also test a few useful features of the DataWarrior package that can help us better analyze chemical compounds. By the end of this short video, you'll know how to calculate the Structure Activity Landscape Index, identify activity cliffs, and apply these insights to guide your hit-to-lead optimization process. Before we dive in, I want to emphasize again that this is a simplified example. In a real-world scenario, preparing the dataset for the analysis usually takes much more effort. You'd need to check for missing values, ensure consistent activity units, remove duplicates, and carefully curate the chemical structure. Activity Landscape Index is a metric often used in cheminformatics and drug discovery to quantify how small changes in chemical structure affect biological activity. It is especially useful for identifying activity cliffs, which are pairs of structurally similar compounds with large differences in biological activity. This method calculates a ratio that compares the difference in biological activity between two compounds to their structural similarity. High values can indicate the presence of activity cliffs and possibly situations where compounds with similar structures show different biological activities. There are some limitations to this method. First, it is highly dependent on the type of molecular fingerprint and the similarity metric used. Second, you need reasonably dense SAR data, not just a large number of compounds, but also a dense sampling of the chemical space, especially around regions of interest. This data requirement can be particularly challenging in typical academic settings, where data sets are often small. This is the equation used to calculate the Structure Activity Landscape Index. The numerator is the absolute difference between the biological activities of compounds A and B. The denominator is 1 minus the structural similarity between the two compounds, usually computed using molecular fingerprints and a similarity metric such as Tanimoto. You could even try to compute SALI values using predicted binding scores from molecular docking, but only if you are confident that your docking engine and scoring or rescoring function perform reliably for your protein target. This is a short summary. High value usually means you have an activity cliff, two molecules that are very similar structurally but show a large difference in biological activity. A low value means the activity changes smoothly across structurally related compounds. Here's a quick example. Compounds A and B are very similar. However, the experimental inhibitory measurements are quite different, which results in a relatively high SALI value, a classic activity cliff. So, how can SALI help during lead optimization? Activity cliffs show us the key substituents or motifs that drive big potency changes. These cliffs reveal SAR hotspots, thus regions where small modifications have a large impact. In areas with low SALI or smooth SAR, we can explore broader scaffold changes. In high SALI regions where the SAR is rugged, we need to make careful, focused modifications, since small changes can have big effects, good or bad. Overall, SALI helps us prioritize series where the SAR is interpretable and optimizable, and deprioritize series that are chaotic and hard to model. This 3D view of the binding pockets shows where activity cliffs occur and, in some cases, helps us explain them. For example, when a specific interaction or steric clash causes a big change in activity, but keep in mind, not all activity cliffs can be rationalized in 3D. Sometimes they result from experimental variability, assay conditions, or even data quality issues. If we, for example, look at the thrombin pocket, in some regions, often deep in the pocket, small structural changes can lead to large activity differences. In contrast, regions that are more solvent exposed are usually more tolerant, allowing for larger modifications without drastically affecting activity, or indeed these regions be used to improve activity. Of importance, when we use machine learning for molecular property prediction, we rely on the principle that similar compounds usually have similar properties. However, activity cliffs are an exception to this rule. These are structurally similar molecules with very different biological activities. Because of this, machine learning models can sometimes severely mispredict the activity of compounds near these cliffs, even if the model performs well overall. Now, let's look at our thrombin dataset, the one we prepared back in session 5. We'll open the DataWarrior file, which contains a set of thrombin inhibitors downloaded from the ChEMBL database. For these compounds, we have inhibitory constants, p-values, and other related data. In addition to the activity values, each compound also has a so-called mol-regno number. This is simply a unique identifier used in the database to keep track of compounds, a bit like a compound's internal ID or name. Just a reminder, this data set is not curated. You should remove duplicates if any, remove salts and mixture, clean the chemistry if needed, and so on. So now we will click on the structure window, thus the filtering area of DataWarrior, and draw a chemical group just like the one shown here. Then we take the lasso tool, select the 3 atoms as shown, and double-click on them. This opens a window where we can tell the filtering tool that these 3 atoms can be in fact any atom, not just carbon atoms. There are many other rules you can apply here, but today we'll focus on using just this simple, customized filtering rule. We began with more than 3,000 compounds, but after applying the structural filter, we're down to just about 42 molecules, depending the database version used and the filtering steps you applied in session 5. These 42 molecules all share this particular structural group. We can now go to Data, then delete rows, and delete invisible rows to remove molecules we do not want to keep for this session. Then we go to Chemistry and compute activity cliff. We use fragFP as fingerprint, but others can be used like circular fingerprints, flexifor, and so on, depending on the data. Then we select for the activity column the one that contains p-value, we leave the default parameters, and create a document that will show the structure pairs. For the identified column, you can select, if you want, a column that contains a key that uniquely identifies molecules, but if not, DataWarrior will create identifiers for you. For the similarity limit to group compounds, you can use the automatic method, which usually works well. You will create a similarity map in 2D, and you can also create a new document in a new window that contains the 2 neighbor molecules and additional columns containing molecule identifiers, similarity, activities, and SALI values. If we look first at the similarity map with this one, we can visualize the structure activity landscape. Here each node represents a molecule. The size of the node reflects the SALI value. The edges connect molecules that are similar above a certain threshold based on the selected fingerprint. DataWarrior also applies some additional adjustments to this threshold, taking into account the neighborhood relationships between molecules. Compounds are also colored according to the activity values. So, when you see a cluster of connected nodes with many different colors, we conclude that the SAR is not flat here, meaning small structural changes lead to large activity shifts. If you move your mouse over a node, you'll see the experimental affinity and the computed SALI value. If you click a node, for instance, one with a high SALI value, you see in the neighbor tree window this compound in the center of the figure and its neighbors colored according to binding affinity values, for instance. Now, using the mouse or touchpad, if you select all these compounds, DataWarrior will display the selected molecules and their neighbors in 2D, with information about the SALI value, the similarity, highlighting some colored substructures. We'll come back to this in a moment. Now, if we look at the other window, with the activity quiff analysis, we have many additional information. We can, for instance, rank the molecules according to SALI value and look at molecules with high value, so we can look at a pair of compounds with high SALI value. But you see that the 2D structures are not shown in the same orientation. This, however, would be useful to compare the molecules. So we go to chemistry, generate 2D conformers to reorient the molecules according to the most central ring system. You can also draw the substructure that you want to use to align the molecules. Now that the molecules are aligned in the same orientation, it's easier to spot the chemical group responsible for the change in activity. For instance, in one compound, we see a modification of the benzimidine group, which leads to a loss of activity. This part of the molecule normally fits into a small subcavity of the binding pocket. Now, let's go back to the main window and focus on just two molecules to make things easier. If we select the two compounds in the neighbor tree window, we see the two compounds, see where are the changes in the structures. We can modify a little the setup to have a better view. I'll come back to the structural differences and explain the meaning of the color codes in a following slide. Now, if we, for instance, want to dock this compound using an external docking engine, we can generate the 3D structures by going to chemistry, then generate conformers. We can keep the default parameters here just to try. You see that one molecule could not be energy minimized. The 3D structure was generated, but some force field parameters were missing to optimize the geometry. In a real project, you want to look more carefully, optimize again the 3D structures, but for now, we leave it as it is. Remember also that we did not perform data curation. Onway, so now we can look at the compounds. We see that in the structure window they are no longer in 2D but in 3D. Now we will save the 3D structures to a file. So we go to File and select Save Special. We want to save the molecule in the SD file, possibly the version 2 of the SD format. We will tell the package we want molecules in 3D, and for the compound name, we can select a column like molregno, and if not, DataWarrior will generate a number. This is it for today. Now we can close DataWarrior. Before we close, I would like to come back on the meaning of the colors when looking at compounds. So here we look at this example where we have only two compounds. The selected compound, which becomes the reference compound, has a red frame. In orange, this is where we have structural differences. In white, this is what is common to both compounds, and in blue, this is what belongs to the actual compound and is missing in the reference compound. From all the sessions we've had so far, we can see that drug discovery is a complex and challenging process. Fortunately, there are many powerful tools and software packages that can help us explore data and support decision-making along the way. That's it for today. We'll continue our journey with DataWarrior in the next session, exploring new tools and functionalities. Until then, take care.